It's my pleasure to introduce Dr. Katie VanValin. Dr. VanValin is our beef extension specialist at the University of Princeton. Uh, Katie joined us again during the pandemic, and she started January 2020. And she's going to share some information with us in regards to, do you know what you are feeding? And with that, Dr. VanValin, I'm going to turn it over to you.
All right. Well, I actually spent some time this afternoon and pre-recorded this talk on the off chance that my toddler did not cooperate, but he did. So I'm able to give this to you live, and I don't have to sit through my listening to my own recording. So this should be better for everybody this way. Um, so we're going to kind of go through tonight a couple projects that we've been working on over the last year, really trying to understand um, co-product feeding, and then also touching on some fescue toxicosis risk for our stocker operations, and of course our any of our cow calf and grazing based operations as well. And then I'll lastly touch a little bit on conserved forage quality, to really try to understand what it is that we're actually feeding. And so when I was in graduate school, this was a kind of a joke, but there's some real seriousness and validity to it. Is that whenever we think about feeding cattle, um, there's really four diets involved. The first one is the diet that was actually formulated. So if I'm sitting at my computer with my ration balancing software out and plugging in numbers, there's what I formulated. Then there's the diet that may actually get mixed. Then there's the diet that gets actually delivered to the cattle, to the pen, to the feeder. And then there's ultimately the diet that animal eats. And I can tell you that there's a lot that can change from the computer and the formulation stage to getting to where to what the cattle are actually consuming. And some of that is human error. Some of that is, just the what the calf decides they're gonna eat, or if they're able to sort through that diet. If we can certainly limit some of that variation that happens between formulation and diet consumption by the animal, then we're going to be better off. And so, a lot of times, when we're talking about diets and diet formulation and rations, we just use kind of quote unquote book values for a lot of our co-product feeds. And so we wanted to kind of do a survey or sampling of different co-products from around the state of Kentucky over a multiple month period, about a 6 month period. And I'm going to share with you some of what we found in there, and kind of how I look at this, and maybe give you guys some tips for handling some of these co-product based feeds. So when I say co-products, that is a broad term given to feeds that are produced from the manufacture and process of another industry. So you look at that list there of feed ingredients, and my guess is that we probably don't have too many cattle in the state that aren't that are being fed, that aren't consuming some type of a co-product. At the very least, you may have some of it in your minerals. So they're widely used, and being that they are quote unquote a waste product, um, they can be economical to feed. But ultimately, as we've kind of developed a more mainstream market for these waste products, that value can sometimes seem to disappear for us when feed costs get a little bit high. But that's when we start talking about co-products. That's kind of the broad term. So it's more than just the products from the distilling industries. It's from multiple agricultural industries as well. Some disadvantages, or maybe things that we need to consider. Disadvantage might be too great of a word. Is geographic availability. So first one that comes to mind is cotton co-products. Up in Kentucky, we don't have very many of those. I work with a few producers that are right on the Kentucky-Tennessee line that can sometimes get a hold of those types of products and make those work. Whereas our friends down in Tennessee have much more availability of some of those types of products, and then obviously as you move further into the south, there's even more abundance of those. Um, Depending on what type of product we're looking at, if there's quite a bit of moisture, there can be transport or handling challenges with some of these products. Availability is a big one. I work with producers that start feeding a certain feed ingredient, and sometimes it's a pretty big part of the diet that they might be feeding, and then all of a sudden that plant shuts down for a couple weeks, or they just don't have a load available that week, whatever the case is, and then you're kind of left scrambling. And so that's something that you need to kind of be aware of and thinking about. And then lastly is this idea of consistency, and we're going to talk about consistency of some of these feed products and what we found from the samples that we pulled. So we worked with county agents across the state that were pulling samples from us or for us on our behalf, and so we were trying to get different co-products from different sources over a like I said, about a 6-month period of time. And so I'm only going to show a couple of these just for timing's sake. But the first one here is dried distillers grains, and so if you look, this first row here is this labeled N is just the number of samples that are included. The mean, of course, is just the average, and then that CV number it's really just giving us an idea of how much variation there was across all of these samples. And then to give you all some context, I wanted to throw in the book value that um, in this case is reported in the nutrient requirements of beef cattle book that was published back in 2016. So that's kind of the gold standard source that we use for all things beef cattle nutrition. So you can see that really dried distillers is pretty consistent with the book values. Maybe, a touch lower on protein and some lower on some of the energy value from the average. But there is some variation. So the higher that CV number, the more variation there is. Um, this is a small data set, and so you get one or two samples that gets off, that can really throw that CV number and inflate it more than maybe if we had sampled 300 or 400 ingredients in this set. So keep that in mind as we're looking through some of these tonight. And then I wanted to kind of go from one extreme to another and look at some more high moisture feeds. And so one of the things that I saw as I was kind of looking at these dry versus wet ingredients, and we'll have some discussion about that, is that moisture content becomes much more variable when we start dealing with wet ingredients. And that can just introduce more variability in that feed stuff. And so I kind of saw that as I was looking through the data set. So on average, honestly, we protein wise are pretty close here, but we do have some differences from from this particular book value. There are other quote unquote book values that people use as well. And then lastly, I wanted to look at stillage or slop. A limited data set here, so I don't want you to read too much into it. But we do have some variation. There was quite a bit of variation, and I apologize. I probably should have put the range on here for that, at least for that fat value. We do see some variation there, and really some of that fat content in something like stillage can kind of limit how much we want to include in the diet, so that we don't end up with too much fat in the diet that can negatively impact things like fiber digestion. Um, the other thing that's not reported here, just because I didn't have enough samples that had the value for it, would be something like sulfur. That becomes something that may limit how much of a certain feed we want to put in the diet, so that we don't run into issues with sulfur toxicity. So from a lot of from what I saw with the dried distillers, we also looked at gluten feed, soy hulls, soybean meal. Like I said, just for timing, I didn't show you all of those values tonight. They appear to be a little more consistent than some of the wetter co-products that we looked at. And so, if we're buying commodity feeds from our local sources that contain dry ingredients, we may not need to test regularly, uh, knowing that a lot of our feed mills, especially if they're formulating rations for people with those ingredients, that they are testing those ingredients most likely. But certainly, if we're switching to a new source of an ingredient, or if that product that we get appears different from load to load, it's probably worth taking sending off a sample and getting it analyzed. There's some, options to get these sent out to commercial labs where you're talking about 15 or $20 to send off a sample similar to what you would do with forage testing. Another thing to kind of keep in mind is that when we start thinking about purchase or mixed feeds, that come with a feed label, there is some level of testing that's occurring just from regulatory services to ensure that those label guarantees are met. Um, Now, keep in mind that that doesn't test for every nutrient that we looked at in this study. For example, you're not going to find a net energy number or a TDN value on that feed tag, mostly because those things are calculated in most instances using an equation, and there's a couple different ways or more than a couple different ways to calculate those sometimes, and so that can introduce some differences, depending on how that value or that energy value is calculated. So you're not going to expect to see that as a guarantee that's on there, but I do just point that out, that if you are, buying a commercially, a commercial feed with the feed tag on there that's got some guarantees, that those are subject to regulatory services. I wanted to touch a little bit on this idea of wet co-products, and really driving home the impact that moisture ultimately has. Moisture in the feed is really just water, and when we have something that's a dry ingredient, we can double that amount of water. So I've done from this beaker to this one here, I've doubled the amount of water. But if you look at as a part of the whole, it's a relatively small proportion. Whereas over here on this side, when we start changing that moisture content, that moisture is such a big factor in the overall content of that feed. And so I wanted to show that to you. I'll show that to you in another graph in a real world scenario as well. So I do think there's some opportunities when you're thinking about feeding co-product feeds. We do want to keep an eye on what that moisture content is, and there can be some more variation. And so I do like the idea of maybe testing those more often than we would some of the dry products. Here's a kind of an example of what can happen if you say you're feeding the ration was formulated at 5.5 pounds of dry matter, and if I assume that wet cake was about 28% dry matter, and that comes out to about 20% as fed, whereas if I'm at 40% dry matter, that's only about 14 pounds as fed. And so um, you can inadvertently either over or underfeed cattle just based on that moisture content. And this graph, I think, does a really great job of showing that. So this is some work I did as a graduate student when I was doing my PhD, and the purpose of this study had nothing to do with moisture content of feed, but this is just one of those things that happened in research. And so these were some growing cattle that were on a mineral study, and they were eating a diet that was about 85% corn silage, so very heavy corn silage diet. And we ran out of one of our sources of corn silage at the farm, and had to switch to a new bag that was harvested under a different set of conditions. And when we opened that new bag, we didn't I say we, I wasn't involved in making those decisions, but we didn't have an updated nutrient analysis on that or moisture content on that corn silage. And so we went back and the cattle were kind of being fed the same amount that they had been the day before. So everyone's kind of cruising along here, and we're looking at dry matter intake, and it's slowly increasing. Each dot on here is a day of intake. And there's some noise in this data set, and that has to do with, I think, somewhat of how we collect this data. But you'll see they are gradually increasing, and then all of a sudden they plummet. And that really was because we realized about a week later that the corn silage in that other bag was 10% wetter than what we had been feeding previously. And so when you go around and you make that same feed delivery, you've ultimately provided them with less dry matter, less energy, less protein. And so that does set those cattle back. And it took us a while. You don't even see it in this graph because it wasn't even during the course of that study that they caught back up. But as they moved onto their finishing diet and transitioned onto a different ration, we were able to kind of catch them back up and they moved on. So this was a mistake, but it's a valuable lesson and something that I don't want to see other people have that same mistake. So we're gonna switch gears here a little bit and talk about fescue toxicosis risk, and so kind of targeting some of our more grass stocker operations, and certainly any cow-calf producers that we may have on tonight. I think this will um, give you all some things to think about. And so when we talk about fescue toxicosis, of course, we start with having fescue-based pastures here in the Mid-South. And so that fescue pasture, that fescue plant, a portion of those are going to be infected with something called a fungal endophyte. So that term means that fungus is inside the plant. So of course we cannot go out and look at one field versus another and tell which one has an endophyte and which one doesn't, or look at one plant from another and know. We would have to do some laboratory analysis. This is kind of a microscope image of the plant tissue there, and you can see that little blue squiggly line is the actual fungal endophyte that's in there. And traditionally, these fungal endophytes have produced a family of compounds known as ergoalkaloids, and the most predominant ergoalkaloid that those fungal endophytes produce is something called ergovaline. And it is that ergovaline that leads us to these whole host of symptoms that collectively we all know as fescue toxicosis. So I apologize, my source wasn't showing up on these photos, but this came out of one of the fescue toxicosis extension publications that we have here at UK. So hallmark of cattle in ponds or in mud holes trying to cool off. Of course, fescue foot there where because the blood vessels constrict as a result of that ergovaline consumption, the blood flow to the peripheries is reduced, and that hoof tissue can become necrotic. And then, of course, the hallmark of decreased average daily gains, which we think is related to decreased time spent grazing on those toxic pastures. So I wanted to find a way to kind of assess the risk of a pasture for causing fescue toxicosis. We know that our pastures are not a monoculture. At the research station at Princeton, we have some um, specific fields that we try to manage in a way that we can induce fescue toxicosis, and we are not the only experiment station or university farm that puts a lot of effort into trying to make sure that we can routinely induce fescue toxicosis as part of our research programs. And so I kind of wanted to do a survey of normal everyday farms, not ones that have either been trying or have been trying to create these fescue toxicosis prone environments. And so what we did is we went out and first tried to determine the botanical composition of that pasture. Like I said, these pastures are not a monoculture. You can certainly see that in that picture there. You do not have to be a forage ID expert to know that we've got some clover in there and some other grasses in there as well. So the way we did this botanical composition, we started with a livestock panel that had 4-inch squares in it, and we cut that down into a 5x5 grid. So there's 25 cells, and essentially you go out into a pasture, place that down in 20 random locations, and you record the dominant species in each one of those cells. And then you can calculate the prevalence of any given forage species in that particular pasture. Next, what I wanted to do is look at how much endophyte is actually out there. So what is the infection rate of the fescue plants in that pasture? The literature would suggest maybe 60 to 80% infection rate. And in full disclosure, that this little part of this project has not been completed yet, so we're going to use some book values if you look at some of this data, but we do have the ergoalkaloid concentrations. And so that's how much toxic compound or ergovaline was actually present in those fescue plants that we sampled. And so the idea here is that if you can take a pasture and you know the percent of fescue that was in that pasture, how much of it was infected, and what the concentration of ergovaline is, then we can start to estimate the ergovaline content in the diet. Because if we just go out and test for ergovaline and we get a really high number, well, then we might think we have a high risk of fescue toxicosis. But if tall fescue was only 20 or 30% of that pasture, well, then maybe our risk was lower than what we thought. And so that was kind of this whole premise, is I wanted to see what was out there in the real world. So when we went through these fields, we did 18 fields at 8 different farms, and some of these were in central Kentucky. Dr. Lemkuhler helped us with this project, and then others were over our way in western Kentucky. And so when we went through and classified those cells and the predominant grass species in those cells, we had fescue, orchard grass, bluegrass, clover, other grasses, weeds, litter would be literal trash in the field, and then bare spots. And so you can see I pulled some descriptive statistics here on these. So we've got kind of the average. And so you can see that fescue certainly dominated our pastures that we looked at. We did have a range on clover. So we looked at, I do have the min and the max here. So you can see these pastures range from 5% fescue all the way up to 94% fescue. And there was some weed pressure in some of those pastures and maybe not in others. And that's something that we'll talk about when we talk about kind of the limitations of these. So when we tested the fescue from all those pastures, the average ergovaline content came out to be about 544 parts per billion. The minimum I put on there is less than 100. And so the reason I put less than 100 is that's the minimum sensitivity of the test that the laboratory runs. So if it's 100 or less, you just get the less than 100. And then the max was upwards of over 1,000. And so you can see I have that same CV value in there that we talked about earlier. So there's certainly some variation that exists across those pastures. So when I do all that math and I assume a 70% endophyte infection rate, and this is again based on that literature that might suggest 60 to 80%. So I pick, the middle, and we'll actually go back and try to get those analyzed so we have real values to plug in there. But if we assume a 70% infection rate, that average ergovaline of the diet drops from 544 to 232 when we account for how much fescue is actually in the pasture. So that's an average of about a 58% decrease, 57.5. So I've talked about what these ergovaline number concentrations are. So to give you a little bit of context of what would cause fescue toxicosis, what levels um, are we looking for? Um, Literature would suggest 100 to 300 parts per billion of total diet may be enough to induce fescue toxicosis, and other studies would show upwards of 750 parts per billion. And so, in talking with some of the folks at our veterinary diagnostic lab, when we start to consider some of the other environmental factors like heat stress, for example, that we know make fescue toxicosis symptoms worse, and kind of as compounds on top of that fescue toxicosis, then the actual concentration or load of ergovaline needed to induce fescue toxicosis may be towards that lower 100 to 300 parts per billion range. There's some limitations though to this approach, and we really don't have a good survey of beef cattle pastures and what is really present in those pastures. And so this was really our attempt to try to start getting some of that knowledge. This is not perfect, and any of my scientific colleagues could probably shoot a couple holes in this approach, but until we have the ability to figure out what exactly it is those animals are grazing, we have to do some estimation and do the best we can. This is some work done by some of our colleagues over in the forages group with Plant and Soil Science Department at UK, where they actually went out and visited the same pasture over a year and looked at the variation that exists in ergovaline concentration. And we've known this for quite some time, that there is some variation, but you'll see that those ergovaline concentrations peak in around May and then decrease pretty quickly. And obviously this might differ depending on where you're at, in the state and in the growing, depending on your growing season, that might be off a week or two. But it decreases really rapidly. And so in full disclosure, we took these samples really any time between May and June. So we may have gotten the peak of one pasture, or we likely missed that peak in some pastures. So we use this kind of as a grain of salt and not a definitive, here's my answer, and I know I'm not going to get fescue toxicosis. I just don't want anybody to read that much into what we're showing tonight. The other thing to think about is that that botanical composition changes throughout the year. What weeds are present? If you have any warm season grasses versus cool season grasses. In talking with my forage colleague there at Princeton, Dr. Teutsch, he said, well, of course, if you went out there today, there'd be no crabgrass. And if you know him, he's a big fan of crabgrass, so he had to put his plug in for that. But he has a good point. And so when we do these types of pasture assessments, they really only hold true kind of for that moment of time in which we sampled them. And then lastly, we're assuming that cattle are going to be grazing those pastures consistently, and we know that that's not true. We know that cattle do have some level of animal preference. And so as another thing is when we look in some of these fields where maybe they had a heavy weed pressure, I think one of them had upwards of 60% weeds, well then we know that the cattle are probably going to are going to be putting more pressure on what grasses are available in that system. So again, it's not perfect, but it does start to give us an idea, at least at that moment in time. And certainly if you could, measure this out over a few months in some of these same pastures, you might get a better idea of your total risk. But I wanted to try to break this down a little bit more. So I categorize these as being low risk, so those that had less than 100 parts per billion, a potential risk at 100 to 300 parts per billion, moderate at 300 to 750 parts per billion, and then high risk would be anything over 750 parts per billion. So the percent of the fields that we looked at, assuming that 70% endophyte infection rate, um, about 40% of them were low risk. About 28% is a potential risk, and then 33% there is moderate risk. And we didn't have any that came in as high risk. Now, like I said, we got those in May into June. So if we were to know the exact day that there was the highest ergovaline concentrations in the pasture and went and sampled on that day, I bet you some of these pastures would have ended up in the high risk category. Just for my own curiosity, and then I put it in here to share it with you all. If we went to the upper end of that infection rate, so 80% endophyte infection rate, now all of a sudden, you start to see that we have a couple fields creep into that high risk category. And we have more fields move from a potential risk to a moderate risk of fescue toxicosis. And so um, there are certainly things that there are certainly pastures out there that present more risks than others. A couple things just to kind of note: a couple of these pastures that we sampled, we knew they were novel endophyte. So novel endophytes are those varieties of fescue that contain an endophyte, but that endophyte does not produce the ergoalkaloids. And so we saw those come back quite low, as being low risk. We saw on certain farms where we knew that some of this fescue had been seeded a long time ago. It was a pretty old stand of about 90% fescue. Came back very high. It was one of the higher values that we had, and so we saw some things in there that made make a lot of sense to us in terms of what we do know about ergoalkaloid levels in fescue. And so, like I said, there's multiple pastures in there that were known to contain some novel endophyte fescues, and they did have they were in our low risk. They tested lower than 100 parts per billion of ergovaline. So the last thing I wanted to talk a little bit about is conserved forages and assessing the value of nutrient content in those. Variation in hay quality exists. I think our forages are much more susceptible to variation just based on how they're harvested, and species that are present can also play a role in there. So this is some data from I believe it's the 2023 Eastern Kentucky Hay Contest. Just looking at the grass hay samples that were submitted there. So I took out legumes in this particular data set. And so up top, we're looking at crude protein, and below, we're looking at energy. And each one of those dots is a sample. So you can kind of see the variation that exists across that data set, looking at both crude protein and energy. And so when we start thinking about that, even in a growing diet, where certainly they're still consuming quite a bit of forage, but maybe less than when we're thinking about like a cow during the winter. That difference in forage quality can impact as much as a quarter of a pound of average daily gain. And that's assuming if I took some really poor quality, stemmy, mature type hay versus something that had a little more quality. So even in these kind of lower inclusion levels, 25-30% of the diet, that hay quality can have an impact on animal performance. We always recommend that people test their hay just because of the variation that exists. And keep in mind, the more forage that's in the diet, the more opportunity you have for variation in your diet because of forage quality. So it's not something I like to guess at. There are book values for hay, but it's kind of funny when I look at what those book values are and compare it to a hay sample that somebody actually sends me. Inevitably the energy or protein is going to be way off. So if I find the book value that matches the energy, it feels like the protein number might be off, or vice versa. So when you call and you say, "Oh, I've just got some average hay," um, you know, that leaves a lot up for interpretation. So things to think about when we're testing, and I say conserved forages because I know a lot of people are using things like baleage and silage and as well. Keep in mind that the results are only as good as the sample that we collected and sent in. So we want to use proper sampling technique and handling that sample properly as well. Kentucky, we have a really great forage testing program. A lot of our county agents will work with you to get those samples collected and sent off. And I know Tennessee has a great network of agents as well that will help support those things. If you are sending them off on your own, there's a lot of different laboratories out there. You can look for ones that are certified through the National Forage Testing Association or NFTA. They will usually put that somewhere on their website. And essentially, that is a third party organization that goes through and we'll submit a known sample to the laboratory to make sure that the results that are coming back are accurate. There's a lot of different options when you send in a hay test, and so this is something that if you don't know, if you've never done one before, you don't know what you're looking at, certainly ask for some guidance on that. You'll see things like NIR or wet chemistry, and you'll see that the NIR samples are cheaper, so you'll probably want to go that route. What I tell people is NIR can be good. It can work really well. NIR is near infrared spectroscopy, and so essentially, we are it's a tool that basically shines a light through the forage and measures that reflectivity, and we have some equations that basically calculate what that light means. And that sounds like a lot of magic, but there has been a lot of work to refine those equations. And so if you are working with a good lab with a relatively known sample, that can work. And what I say by known is that the equation to predict energy in something like grass hay versus a legume is different. The equation that they would use for mixed samples is different. And so if you start getting into anything that's a real mixture of things, or maybe some odd ingredients, then wet chemistry is probably the better route to go. It is a little bit more expensive and it may take a day or two longer to get your results, but it can be a more accurate way to measure some of those ingredients that may be a little different. The other advantage I like with wet chemistry is for any of our fermented feeds, we can add on a fermentation panel to those wet chemistry analysis. And all that is doing is it looks at the pH and it looks at the acids that were produced as a result of that ensiling process, and can give us some indications of how well the forage ensiled, and that can give us some clues if we suspect there might be a clostridial fermentation, which can lead to botulism, or we can see that maybe the ensiling process was not as good as we'd like it to be, and maybe that may have some impacts on forage stability and things of that nature. It can kind of just give us an idea of how well that ensiled process goes. So I do try to recommend that for folks that are testing any of their fermented feeds. So to kind of wrap all that up, I know we covered a lot of topics there kind of quickly, but um, our co-products can be really economical feeds, but be aware of things like availability and transport. Our high moisture feeds seem to be a little more susceptible in variation, and that's really just due to moisture content alone. And I didn't even touch on moisture content once it's gotten to your farm. And there's been instances where we have some of these piles that get rained on or things like that, and that certainly can add some moisture to it that we may not be accounting for in that formulation. Um, and then switching gears on our fescue toxicosis, if you suspect that you've had issues with fescue toxicosis, that might be worth assessing those pastures. If nothing else, doing the botanical composition can really give you a good idea of what species are out there and prevalence of weed pressure, and really kind of help you understand a little bit better what's actually in your pasture and once you kind of get the hang of it, you can go through there pretty quickly. And if you look through some of the other extension publications, there's other ways, other methods of doing pasture botanical composition that you can kind of do as you're going out through your pastures, maybe rotating cattle or those kinds of things as well. And then lastly, just touching again on the stored forages, there's a lot of variation that can occur there. And so understanding what that nutrient content is can go a long way in helping reduce the variation from formulation to feed consumption by our cattle, and try to make our feeding programs a little bit more accurate along the way. So with that, I am happy to take any questions that you all might have.
Great job, Dr. Van Valen. And so there was one that came in about the utilization of red clover to help mitigate the fescue toxicity and toxicosis issues.
Yes, that's some really exciting work. I feel like every time I hear a new update on that work, it gets more exciting. And so I also will put a plug in that if you're interested in kind of a deep dive on red clover and fescue toxicosis, that Dr. Brittany Davis with USDA ARS out of Lexington was at the beef efficiency conference last month, and that I know that video's out there on YouTube, and she does a really great job. So I'll try to briefly summarize the breadth of work that she's been doing over the last couple years, and her predecessors before her. So red clover specifically produces a compound that seems to mitigate fescue toxicosis when they've tried to feed red clover leaf in a mineral. So they feed up to like an ounce per head per day, I think is what it is. It's like 25% of a 4 ounce intake mineral, and see uh, complete amelioration of fescue toxicosis. So they look at that blood vessel constricting, and that seems to go back to normal. They have yet to find a bottom, so like a minimum amount of red clover that fixes that, or that would prevent that. So it's very intriguing. It's very interesting. For years, we've always thought about clover being a dilution effect when we put it in the fescue pastures, but it appears there may be more to it than that. And I'll just touch briefly because it's always the follow up question is, what about white clover? Can I put ladino clover? That's a lot easier to get established. And um, the folks that are working in that area will tell you that those other varieties of clover produce some of that compound, but not nearly as much as what's in the red clover. So I know there's some other work being done with some soy products that things like soybean meal that may have some of that content, and I think they're actually maybe adding that to some of the forage variety trials this year, where they look at different varieties of clover to test and see which varieties may have more or less of that compound. So um, I would stay tuned on that, but they have a lot of really compelling data, good compelling data that suggests that effect is real. It's just a matter of getting it into the cattle in a way that is easy and practical to do.
So when we look at co-products, and you showed that on the wet feeds there can be quite a bit of variability. And for us, one thing, be it the, we got bourbon and fuel ethanol distillers coming in. Did we make note of whether those were fuel ethanol versus bourbon ethanol distiller samples?
We did not, but I do know that most of those dried samples came from the central Kentucky region, and I don't think very many of them came from over my way, which is where the ethanol plant, fuel ethanol plant would be. Um, I do know on one of the stillage samples, one of those came from a rye run versus a normal corn run. But when I pulled that one out separately, it really didn't look very, very different. But I know that's a question I've gotten from folks that are using those, because there's a kind of a odor difference when that gets delivered. So there's been some question about what the nutrient difference was, but I didn't didn't pick up a big difference from the average when I looked at that.
Anybody else have any questions? You can type them in the Q&A box. I don't see any in there right now, Dr. Van Valen. I do have one quick question for you that we seem to get all the time, and it's in regards to the fescue toxicosis and the ergovaline levels. And the levels, we think about a lot of hay being produced, right? And so when we cut that forage and we're going to make hay out of it, do those levels stay the same as what's in the pasture, or do they decline as that hay goes through its curing process?
Yeah, we can still see high levels that show up in the hay. In fact, I think we've had a few cases here recently in the state of fescue foot. And we always talk about fescue toxicosis and heat stress, but if you think about cold stress, that can impact the blood flow to the peripheries and make that fescue foot worse. And I think some of those cattle were actually consuming endophyte infected hay, if I remember. It's not that they were just out on stockpile or something like that. So that is still something to kind of be aware of. And um, how where on the plant you look at. So the seed head is where a lot of that content is. And so if we get some of that really mature hay that's got seed heads in it, that can also impact how much is in there.
Yeah, and I think I just wanted to bring that up because we know that probably at least 3 or 4 cases that we've been called in on this winter already, and normally it's maybe 1 and maybe 2 a year, but this year for whatever reason it seems like it's worse, and I can't help but think that part of that might be just the growing season for producing hay, that people got out in the fields a little bit later and plants were maybe a little more mature.
Yep. Yeah, it has felt like there's been an uptick in fescue foot with, and it's kind of coincided with some of these real cold spells that we've gotten. So certainly as we get ready to go into another one, certainly something to be on the lookout for.